And first and foremost, I would like to welcome Dr. N. Haraprasad sir for accepting our invitation to deliver today's session on recombinant DNA technology. Before starting today's session, I would like to give brief introduction about sir. Dr. Haraprasad N. sir is a self-motivated achiever with PhD in biotechnology from the University of Mysore. He has 28 years of professional experience in teaching, research, and administration by incorporating research goals through academics with sustainable and organizational partnerships between different organizations. He is professor in the Department of Biotechnology at JSS Science and Technology University, Mysuru. His subject expertise gives in ease to fill in the classes and troubleshoot any objective-driven assessments. His research interests include plant pathology, screening of antiviral compounds, medical biotechnology, biosimilars, signals transduction in metabolic disorders, and he has supervised 47 UG and PG students for their dissertation work and presently guiding one doctoral student. One of his doctoral students has successfully completed his PhD program and he has to credit 39 research publications, one published book, and has co-authored one book chapter. He is a strategic leader with a proven track record of building and leading effective cross-functional curriculum and driving change initiatives at all phases of academics. He is a proficient problem solver with analytical skills and necessary for today's academician. He is a proactive agent and spends most of his time in student environment, encouraging, learning, motivating, and promoting to bring the best out of them by pointing their strength and weakness in appropriate manner to bring in value-added results. Not only students' personal level, but also academically. He is a zealous orator and has delivered many workshops for students, researchers, professionals, and academicians. He is an outstanding academician and administrator also who coordinates and communicates between fast-paced students and highly skilled professionals. He has traveled abroad on scientific assignments on several occasions. He has served as an active member of several academic and professional committees within and outside the university. In his stint of 2 decades at JSS Science and Technology University, he has planned and coordinated more than 198 programs. Being one of the pioneering members of formation of the university, he has initiated policy development and creation and various guidelines and regulations in accordance with statutory requirements of state and national agencies. He practices studying current best practices and professional developments with utmost detail and have relentlessly worked towards translating it into relevant programmatic actions for sustainable development of the organization. He has worked as the first deputy registrar of JSSTU for 5 years and have relentlessly served the organization. His objective is to be a loving teacher of simple truth, to teach, inspire, and empower himself and others as well. With this brief introduction, I welcome you once again, sir.
Thank you. Thank you.
Thank you very much, sir.
So, shall I continue now?
Yes, sir. Yes, sir.
We start.
Now the
Yeah.
floor is yours, sir.
Thank you.
Yes.
At the outset, I thank the whole team of KSTA for inviting me, and I also thank all the students who are participating in this program. So it's a pleasure always to be associated with an organization like KSTA, where all vibrant individuals are there, inviting and exposing all the youngsters to such important topics in the area of different science, technology, and culture. So thanks to the chairman, thanks to everybody at KSTA. So today we will talk about a very popularly known technology which is referred and heard of as recombinant DNA technology. So I'm Dr. Haraprasad and I work as a Dean Quality Assurance and Accreditation at JSS Science and Technology University. I also thank my organization and the authorities of the organization of JSS Science and Technology University, Vijay Chamara General College of Engineering, for encouraging me and likewise all our faculty to participate in disseminating scientific and technological developments to the world, especially the young generation like you all. So to move on, let me start telling you about what I would like to present today. So my whole presentation will have a small introduction on the topic of recombinant DNA technology. What discoveries led to this technological development called today as recombinant DNA technology? The timeline, recognizing the timeline of development of recombinant DNA technology. And next, we will try to little bit learn about what is the process of generating a recombinant DNA. And lastly, the applications of recombinant DNA technology. And I also have a surprise for you in the end and related to the present day world wherein we cannot do without that technology which is driving us crazy. So we will move on to the introduction. Before I start telling you about recombinant DNA technology, let me quote this. And it is highly meaningful, especially for the younger generation, to concentrate on every highlighted word here. The past gives us knowledge and experience. The present gives us the power to change them. Together, the past and the present allows us to envision and shape the future. This is what the younger generation should actually look towards in developing themselves and seeing themselves placed in a better world than what you are today. How do you do this? You derive all the knowledge and the experience which your ancestors have developed for you, and putting your knowledge into it, your thoughts into it, your ingenuity into it, you can develop it to such an extent that you will have a better and a brighter future for everybody to live in. So, let us first start with the concept of how the world recognizes something called as a developed country. A developed country is actually having some features which the world recognizes and also relates a developed country with. So just to look at how a country is highly developed. A country is considered highly developed. A country is considered highly developed if it has the following. That is breakthrough remedies for major diseases like cardiovascular disease, cancer, viral diseases, even bacterial diseases, some of one of which we have already experienced now. Abundant availability of clothing, food, and drinking water. Sound knowledge and practice of good hygiene, high quality jobs, technologically advanced industry. And if a country has all this in abundance and also caters to development of every area in such an aspect, then it is considered as a developed country. And the best part here, if we look at all these factors, all the above areas, recombinant technology has a role to play and makes a great contribution in developing each of these areas, which a country gets recognized as a developed country. So what do you see on the left side image? Nothing but sheep, right? So, but what is different about the sheep? They look more healthy, they look alike. All the three are looking the same. So, how does this happen? So, we all say our features, our facial features, our looks depends on what molecule in us we refer it back to in science called as the DNA molecule. So, the DNA is the molecule of the life. So, there are trillions of cells in our body or in the body of any living organism. And we as humans has 46 human chromosomes in each cell, 2 meters of DNA, 3 billion DNA subunits. That is the basis as you have learned it at ATCG, and approximately around 30,000 genes code the protein that perform most of the life functions within our body. Why are we looking into the basic concepts of DNA when we are talking about recombinant DNA technology? How do you relate the picture on the left and the description or the picture on the right side? So, the application of technology to improve a biological organism, that is, the knowledge we have gained from the DNA molecule and the application of technological development to modify what you look on the left side. Very beautiful sheet here. So the application of technology to modify the biological function of an organism by adding genes from other organisms which has features to better the performance of that organism wherein the new DNA has been inserted. This kind of technology, which has developed over a period of time, is what we all today know as recombinant DNA technology. And you should all be knowing what the left side picture, what is that sheet called as. So if you have heard of it, it is Dolly, isn't it? So there are different skills like recombinant DNA technology includes, which has revolutionized one area which we all know as biotechnology. So what are these skills you can also pick up as student later and higher studies if you pursue? These can lead you to working in the area of recombinant DNA technology. For example, as simple as microscopy and imaging. Microscopy you all know, but that enhanced technological developments which has led to the advancement of microscopy techniques and imaging has actually contributed towards recombinant DNA technology. And the very popular technology called as PCR technology, polymerase chain reaction technology, which brings in the greatest power in the hands of mankind to multiply what we just learned as DNA molecules and have as many copies as we want of a particular sequence of DNA and use this, our knowledge, to modify this DNA in generating something which we all want for the betterment of the mankind. So, in this effort, where did we land ourselves? So it is just not enough to have or multiply the single or a kind of DNA, but we should also learn or we should also learn to simulate or actually imitate the way the DNA behaves in life forms. That is generation of proteins. So how did we do that? That was done by the mankind by actually bringing in a technological development which is today known as protein and DNA chips and microarrays. Here in this technology, we see that the small strands of DNA are actually immobilized on, to make it very simple, immobilized on a surface which maintains it in the life form. And when the whole process can be performed on this DNA and generate the proteins we want or any biomolecule we are interested in. Now, the man's mind doesn't stop, so it always ventures out to find new things and ways to do these new things. So we wanted to integrate the knowledge of computers to the life sciences. So hence we developed an area called as bioinformatics. And this skill of bioinformatics has enhanced our capability to actually virtually see what can happen in reality, simulate the real situation. So bioinformatics led to a greater development in the recombinant DNA technology area and is today a very popular skill set required for a biotechnologist and a scientist who is working in the area of recombinant DNA technology. Excuse me. Now, with all these skills and tools, we were able to develop recombinants and monoclonal antibodies to just to refer you in a different direction of required protein which can solve different kinds of problems which mankind is facing. And we were also able to, utilizing this recombinant DNA technology, we were also able to sequence the proteins, nucleotides, etc. There are certain technological developments which has led to better understanding of the way the DNA and proteins behave. That is the flow cytometry and cell sorting technique, the chromatography and electrophoresis, and also to a very great extent the LCMS and multi-top technology, which has led to a better understanding of all those biomolecules which are helpful in the process of recombinant DNA technology. So recombinant DNA technology is One of the recent advances in biotechnology, which was developed by two eminent scientists. We can never forget them any point of time now and in future. Boyer and Cohen. And this happened in the magical year 1974. So here is both Boyer and Cohen. Stanley N. Cohen, born in 1935, received the Nobel Prize in Medicine in 1986 for his work on discoveries of growth factors. How both of them, the smile is actually very satisfactory, them and for us, because both of them have contributed to such an extent to the area of biotechnology that the world can never forget. Herbert Boyer, 1936, he's the one who constructed the first DNA using bacterial DNA in plasma. So together, they have transformed the way scientists look at life and how the magic molecule DNA can be utilized for various aspects to modify life or to understand the life in a better manner. So recombinant DNA technology or recombinant DNA, as it is popularly abbreviated as rDNA, something which we have to understand very precisely before we go into the technological aspect. So what is a recombinant DNA? Recombinant DNA is nothing but recombining the genetic material of different organisms originating from different species together. Is it really possible? Yes, it is, because we are in a world which has already mastered the recombinant DNA technology. So to say in other words, recombinant DNA is actually a DNA constructed using laboratory techniques like genetic recombination, such as molecular cloning, transferring selected fragments of DNA into the other to bring together genetic material from different sources, that is, from different species, and in an effort, creating sequences that would not otherwise be found naturally in a gene. So what we are doing here is bringing in the best part of that DNA sequence present in different species or different organisms together, expressing them, making it functionally viable, and then venturing out into what it actually can generate. And looking into that, we are directing that effort of generating new biomolecules our towards solutions for the challenges faced by mankind. So recombinant DNA technology only becomes useful when that artificially created DNA has the capability to reproduce, and this quality of that modified DNA or artificially created DNA is called as DNA cloning. So recombinant DNA is possible because DNA molecules from various organisms actually share the same chemical structure. Yes, as we say, the blood is red, irrespective of from which species we draw it from. Similarly, the DNA is same, irrespective of from which organism we take it from. So they differ only in one concept, that is the nucleotide sequence within that identical overall structure. So recombinant DNA is the general name for that piece of DNA that has been created by combination of at least two different strands of DNA. Recombinant DNA molecules are sometimes called chimeric DNA. This name because they can be made of material from two different species, like the mythical chimera. rDNA technology utilizes palindromic sequences and leads to the production of sticky and blunted. You understand what a palindromic sequence is? That sequence which reads the same from either side when you read. So that said, and understanding what recombinant DNA is and how the technology which was based on this thing, this molecule formed by combining DNA from different species has led to, let us look into how this all happened over a period of time. Way back in 1941, we see the term genetic engineering was first coined. Later, in 1950, Esther Letterberg discovered the lambda phage. and isolated the lambda phage DNA for manipulations and understanding the functionalities of DNA better. In 1952, we see that first observation of the modification of viruses by bacteria was uncovered. And 1957, again the magical year I call it because this was the first time when synthesis of DNA was made in a test tube, that is outside the living system. 1974, what we are talking about, Boyer and Cohen was successfully able to present to the world recombinant DNA, and recombinant DNA successfully reproduced in Escherichia coli. So, when we have learned what recombinant DNA technology is, in brief, recognize the timeline of development of this technology, let us see a little bit deeper as to what is the process in generating this recombinant DNA. So, it contains 6 steps. First step being isolation of the genetic material from the identified organism. Then, when we isolate it, the DNA is there in our hands. We have to combine it with another DNA which we have isolated from 2 different species at least. So, in order to do that, we have another technological tool which we use called as the restriction enzyme digestion. How we do it, we'll just learn it in a little more time. Then, that's just not enough. I told you about a technology called as PCR, polymerase chain reaction. This technology is used to multiply the amount of DNA we have after recombination. Then ligation of DNA molecules, we put these two DNA together, insertion of recombinant DNA into a host because you cannot keep generating or keep activating it outside. Then isolation of the recombinant cells hosting this DNA. Now, let us understand step by step. First step I told you, wherein we do have a sequence of reactions leading to the isolation of T. And the whole process is explained here in the previous slide as I showed you stepwise. Isolate DNA from 2 sources, cut both DNAs to the same restriction enzyme, mix the DNA, they join by base pairing and add DNA ligase to seal the DNA with covalent bonds, put plasmid into bacterium by transformation, and clone the bacterium to give you bacteria, multiple cells of bacteria having the same fragment of DNA which is recombined. So let us, after that pictorial representation of how sequentially the process takes place, the first step I told you is isolation of the genetic. So in this first step, the desired DNA in its pure form, that is what we mean to say when we say it is pure form, is it has to be free from other macromolecules. You all know when we have to isolate DNA, we break open a cell. So when we break open a cell, there are other cellular components which are also there. We do not want all those cellular components. We just want the DNA for us to go further. So since DNA exists within the cell membrane, along with other macromolecules such as RNA, polysaccharides, proteins, some lipids, and all enzymes also included, lysozyme, cellulase, chitinase, ribonuclease, etc. Other macromolecules has to be separated out. And this is done by utilizing enzymes or other treatment. Ultimately, the addition of ethanol into this solution of into the solution containing DNA causes the DNA to precipitate out as fine thread. And this fine thread is easily looped out to give a pure form of DNA. Now, once we have the DNA, we transfer it into another fresh tube, another fresh and clean tube. And here, what we do is the second step, that is restriction enzyme digestion. Restriction enzymes, as you have all learned, acts as a molecular scissors that has the capability to splice or cut the DNA at recognized, identifiable, specific sites. And such reactions, which happen in the presence of restriction enzyme, is called as restriction enzyme digestion. Process of restriction digestion involves incubation of the pure DNA with selected restriction enzyme at conditions which are optimal for the specific enzyme. So what are these conditions? You add buffer, you create an atmosphere with in a solution wherein the enzyme can act very precisely and also has the capability to cut the DNA at specific sites as we want it to, and then we want to isolate the sliced out DNA. So this technique, called as agarosol electrophoresis, is the one which we use to reveal the progress of restriction enzyme digestion. And using this technique, which involves running out the DNA in an agarose jet and an application of the current, the negatively charged DNA, as you all know, DNA is negatively charged, and this DNA, which is negatively charged, travels to the positive electrode and thus gets separated out based on the size of the fragment. This capability of separating out the DNA based on the size allows us to cut out the digested DNA fragment. So the vector DNA is also processed using the same procedure. That is, from a species, we want to introduce it to another organism, so you isolate the vector DNA also using the restriction enzyme digestion. Now, I said cutting the DNA using restriction enzyme. The restriction enzyme, for example, EcoR1, cuts a circular DNA molecule bearing one target sequence, resulting in a linear molecule with single standard sticky end. I'm not going to dealing with what a sticky end and a blunt end is, what you will understand in a different manner. So, as the depiction shows here, you have two strands of DNA here, AATTC and TTTAA, which wherein it cuts at both ends. So in this pictorial representation, this is how EcoR1 works on the site. So EcoR1 actually recognizes the AATTC site and cuts the DNA at that site. So both running from 5' to 3' and 3' to 5'. So this forms two strands of DNA with sticky ends at both 5' ends. It has AATT and AATT ends which are sticky, that is ready to acquire or get attached to another strand of DNA which is complementary to it. The next stage is to multiply. We only have a few strands of DNA now from the isolated DNA strand. So we use a very important technique called as PCR technique, that is polymerase chain reaction technique. PCR is a technological advancement of making multiple copies of a DNA sequence using the enzyme called as DNA polymerase, and this can be done in vitro. This technique helps to amplify a single copy or a few copies of DNA in thousands and millions of copies, which we can have for further experimentation. When we look at this technique, the PCR reactions are run on thermal cyclers using the following components. What are thermal cyclers? Thermal cyclers are instruments which have the capability to actually provide us with an environment of different temperature conditions within very less lag of time. The first one, which is required, is the template DNA. That is the DNA which we have isolated from different organisms and the vector DNA. And this is the DNA to be amplified. The second requirement is called as a primer. A primer is nothing but a small chemically synthesized oligonucleotide that are complementary to a region of the DNA which have cut out using restriction enzyme. The third, very essential DNA polymerase, an enzyme which has the capability to multiply DNA. Fourth, the nucleotides. Nucleotides are needed to extend the primers by the enzyme. So the cut fragments of the DNA can be amplified using PCR and then ligated together, brought together with the cut vector. And further, we go on to the fourth step, that is the ligation of DNA molecules. What do we understand by ligation? Bringing the DNA molecules together. The purified DNA and the vector DNA of interest are the ones which are cut with the same restriction. Why same restriction enzyme? Generating the same kind of sticky end with complementary features. This gives us the cut fragment of DNA and the cut vector that are now open, and the process of joining these two pieces together using the enzyme DNA ligase what is referred to as the process of ligation. Why do we need to do this? We need to do this to render the DNA molecule functional. So the resulting DNA molecule is a hybrid of two DNA molecules, the interest molecule and the vector. The terminology of genetics, this intermixing of different DNA strands called the process of recombination. This new hybrid DNA molecule is also referred to as a recombinant DNA molecule. And the whole technology which you have seen unravel from step 1 to 4 is referred to as the recombinant DNA technology. But still we are not there. I said there are 6 steps. What is the next step? The next step is we have to see that this is recombinant DNA is delivered into a host so that it can functionalize. So in the fifth step, recombinant DNA is introduced into a recipient host cell, which is mostly a bacterial cell. And this process, as you all learned, is called as the process of transformation. Bacterial cells actually do not accept foreign DNA easily. So what is done? They are treated to make them competent to accept new DNA. And this process of transforming a bacterial cell into a competent cell is known as is what is carried out in this step. So the process for delivering a cell into a competent cell from a normal cell is used. Actually, the process used may be thermal shock, calcium ion treatment, electroporation, etc. So that we develop a competent cell. So upon this, the sixth step is isolation of recombinant cells. Transformation process generates a mixed population of transformed and non-transformed cells. And when we have a mixture of transformed and non-transformed cells, we will have to isolate the cells of our interest, that is transformed cells. So the selection process of recognizing and isolating the cells which have got transformed or filtering the transformed host cells only is carried out. So for isolation of recombinant cells from non-recombinant cells, we need something to identify that cell which is recombined or which has a recombined DNA. That is carried out by something called as a marker gene of plasmid vector, which is employed here in this technique. For example, if you take the PBR322 plasmid vector contains a different marker gene. That marker gene allows us to isolate those cells which has PBR3, which has been transformed. That is, this marker gene is nothing but an ampicillin resistant gene and tetracycline resistant gene. So here, when PST1 recombinant enzyme is used, it knocks out ampicillin resistant gene from the plasmid so that the recombinant cell becomes sensitive to ampicillin. So that is how the recombinant cell or rDNA is generated in steps. And they are also separated out based on its capability of having a strain using a marker to identify and isolate the recombined cells only hosting the recombinant DNA. So with that technology known, let us understand why are we doing all this? Why is it that this technology we should learn about, or what is this technology done to the area of scientific development and technological development in the world? There are innumerable applications of recombinant DNA technology. There is no stopping. You can do anything you think about. You, as youngsters, can actually look into this as an open door wherein you can bring in anything and realize whatever can today be called as scientific fiction. Recombinant DNA technology is widely used in biotechnology, in medicine, in research. What is that we have been able to achieve using this technology today? We are able to survive. The world has been able to survive because of this technology today in the recent experience of pandemic what we had, because this is a technology which helped us generate vaccine in such a short period of time and in such a large volume so that world can survive by using this one. Recombinant proteins are the ones which are widely used as reagents in laboratory experiments across the world. There are practical applications of recombinant DNA which are employed in industry, in food production, in various different agricultural processes, in industrial processes of synthesizing different molecules, synthesizing different biomolecules, etc. Recombinant DNA technology is also used to detect the presence of not only HIV, but this is used as a technique, as a tool for diagnostic, for actually detection techniques, pathological detection techniques of various different viruses and other organisms, because nothing can be as specific as a recombinant DNA. We are very sure what is the sequence of DNA we have introduced, we are using, hence its complementary sequences can be easily identified. So there are different detection techniques which have been discovered and also made available for detection of various biological organisms and other biomolecules by using recombinant DNA technology. Recombinant DNA technology is also used to identify, map, and sequence genes and to determine what are the functions associated with these genes. Recombinant DNA technology finds huge amount of application, a large amount of application in the area of medicine. Insulin production was one of the very popular methodologies using recombinant DNA technology, which was there. And another very interesting area which recombinant DNA technology has led us into is gene therapy. There are various diseases which we have come across which has no other solution, and gene therapy is the only hope. This technology is used as an attempt to correct the gene defects, especially when people are born with genetic defects. There is no medication for that. You will have to go into the DNA level, cut it out, cut out the DNA which is not performing well, and insert such DNA which can actually render this flaw into generating the required kind of biomolecules and thus lead to a normal life of a human being. So this is used as an attempt to correct the gene defects which gives rise to hereditary disease. And the next one, but not the last one at all. There are, I told you, innumerable number of applications of recombinant DNA technology. I have been able to list out here only a few. There is no stopping. You can use recombinant DNA technology in anything. Clinical diagnosis. ELISA, for example, utilizes recombinant DNA technology in a to a very great extent in diagnosing different diseases. So, what has this led us to? Interesting things in the area of science, research, etc. Production of transgenic animals, for example. What is a transgenic animal? We have studied recombinant DNA technology until now, and if you see, genes of interest and beneficial to mankind are the ones which are actually harped upon by the mankind. So this we now, after understanding what recombinant technology, DNA technology is, we can isolate a DNA of our interest, and this can be inserted into animals of again our interest to produce what we call as transgenic animals. The beginning, I showed you the picture of Dolly. So that is one such thing. So it helps. What does this transgenic animals help us in? It helps with the production of better farm animals so as to ensure more commercial benefits. It also ensures production of certain commercially important proteins, pharmaceutical compounds in such transgenic animals, and by technologists have successfully produced transgenic pigs, sheep, rats, cattle, etc. Again, your transgenic sheep. So what? What does this transgenic animals do and capable of? Transgenic cow actually can produce 13% more milk protein than normal one. And the transgenic fish can grow up to 6 times faster than the wild type fish. How much beneficial it is for the mankind. We can actually have a solution. We actually have a solution here for food safety. Tracy is the first transgenic animal to produce a recombinant protein in her milk. Then Enviropig is a transgenic pig which was developed. So all these are transgenic animals with benefits for the mankind. We have not stopped there. We have tried to establish this technology and see how in agriculture we can have higher production and other uh, benefits from developing transgenic plants. Tools and techniques of genetic engineering, recombinant DNA technology are effectively applied to produce transgenic plants, or you also refer it to as genetically modified plants. Many transgenic plants have been developed with enhanced desirable traits. What are the enhanced desirable traits which we want to look at? Where we face problem in agriculture, we want to generate a transgenic plant with overcoming that problem, overcoming the challenge. So plants which are resistant to herbicides, which are resistant to insects or even viruses, or which can express maize sterility. Production of commercially important biochemical pharmaceutical compounds. In fact, recombinant DNA technology scientists are also venturing out into something called as an edible vaccine. Today, you all took a jab to get that vaccine inside your body. But scientists are also trying to manufacture plant parts which can be just consumed, but it harbors the vaccine and delivers the vaccine into your body. So, what picture do you see here? Beautiful white patches on the left picture, and not so good. Not this right picture doesn't make us feel so good. It is dark, providing a picture which shows not all that all the right things are happening on the right picture. But what exactly is this? This is nothing but cotton. Both the fields you see here, both the pictures are pictures of cotton fields. What is the difference here? On the left side, you see that you have a field which is actually flourishing with cotton grown to a grown in a good manner. On the right side, a similar kind of field which was affected by disease by an insect called as cotton bollworm infected, and then the whole crop is destroyed. That is the picture on the right side. So this is the capability of transgenic plants. So imagine the amount of benefit that farmer who had used BT cotton to produce cotton and what would be his outcome. Now, from animals to agriculture to what we all have experienced that can transform the way we live and also assist us in just living and not succumbing to various diseases like the pandemic. Recombinant vaccines. A number of vaccines have been produced biologically utilizing recombinant DNA technology. The present one also, COVID. So these vaccines are effective against numerous serious diseases caused by bacteria, viruses, or even protozoa. They include vaccines for polio, malaria, cholera, hepatitis, rabies, smallpox, COVID, the list goes on. I cannot put all of them here, just so I did put COVID and end here. So the generation of DNA vaccines has revolutionized the approach of treatment of infectious diseases, including the present COVID we all experience. That was just for approaching the challenges. But we need not stop there. We can go forward. Recombinant hormones. Using recombinant DNA technology, bacterial cells like E. coli can be transformed for production of different fine chemicals like insulin, somatostatin, somatotropin, and pre-endorphin. Human insulin hormone, that is humulin, is the first therapeutic product which was produced by the application of recombinant DNA technology. Interferons are glycoproteins which are produced in very small amounts by the virus infected cell. Interferons have antiviral and even anti-cancerous properties. by recombinant DNA technology method. Here we see that the gene of human fibroblasts, which produce interferons in human beings, is the one which is inserted into the bacterial plasmid, and these genetically engineered bacteria are cloned and cultured so that the gene is expressed, and we have interferons which are produced in fairly high quantities, which can be used in various therapeutics. Engineered antibiotics. Antibiotics produced by microorganisms are very effective against different viral, bacterial, or protozoan diseases.
ಹಲೋ ಹಲೋ ಸರ್ ಹಲೋ ಹಲೋ ಸರ್ ಆಲ್ರೆಡಿ ಯು ಆರ್ ಇನ್ ಲೈನ್ ಸರ್ ಹಲೋ
Sir, any? Hello?
ಉಮೇಶ್ ಸರ್ ಅವರು ಲೈನ್ ಅಲ್ಲಿ ಇದ್ರು ಕೂಡ ಅವ್ರದ್ದು ಏನೋ ಕನೆಕ್ಟಿವಿಟಿ ಸರಿಯಾಗಿರ್ಲಿಲ್ಲ ಬಟ್ ಈಗ ಪುನಃ ಅವರು ಎಕ್ಸಿಟ್ ಆಗಿದ್ದಾರೆ
Okay, okay.
ಮೋಸ್ಟ್ಲಿ ಜಾಯ್ನ್ ಆಗ್ತಾ ಇದಾರೆ ಸರ್ ಅಗೇನ್ ಒನ್ಸ್ ಅಗೇನ್
Okay, ತೊಂದ್ರೆ ಇಲ್ಲ. Sorry for the inconvenience caused. At times, network issues. It happens in all the colleges. Meanwhile, if any questions, please do mention in the chat box. You have a good opportunity to speak with all the experts here.
Sir.
You can try to raise your virtual
Sir.
hand so that we can unmute. Please tell me.
ಸರ್ ಇಲ್ಲ ಈಗ ಎಕ್ಸಿಟ್ ಆಗಿದ್ದೀರಿ ಸರ್ ಈಗ ಎಕ್ಸಿಟ್ ಆಗಿದ್ದೀರಾ ಹಾ 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 ಪ್ಲೀಸ್ ಸರ್ ಒನ್ಸ್ ಅಗೇನ್ ನೀವು ಹೆಂಗ್ ಜಾಯ್ನ್ ಆಗಿದ್ರೆ ಅದೇ ರೀತಿ ಜಾಯ್ನ್ ಆಗಿ ಸರ್. ಸ್ಟೂಡೆಂಟ್ಸ್ ಆಲ್ರೆಡಿ ಇದಾರೆ ಸರ್, ಏನ್ ತೊಂದರೆ ಇಲ್ಲ. ಪ್ಲೀಸ್ ಸರ್, ಪ್ಲೀಸ್ ಪ್ಲೀಸ್. ಉಮೇಶ್ ಸರ್ ಇದಾರೆ, ಲೈನಲ್ಲೇ ಮಾತಾಡಿ ಸರ್. ಇವಾಗ ಜಾಯ್ನ್ ಆದ ತಕ್ಷಣ ನಾನು ಕನೆಕ್ಟ್ ಮಾಡಿ, ಲೈನಲ್ಲೇ ಇರಲಿ ಸರ್, ಲೈನಲ್ಲಿ ಇರಲಿ, ಮಾತಾಡ್ತಾ ಇದ್ದೀನಿ.
ನೆಟ್ವರ್ಕ್ ಇಶ್ಯೂ ಕ್ಲಿಯರ್ ಆಯ್ತಾ ಇದೆ?
ಇಲ್ಲ ನೆಟ್ವರ್ಕ್ ಇಶ್ಯೂಸ್ ಏನಿಲ್ಲ ಸರ್, ಒಂದ್ ನಿಮಿಷ. ಇವರು ಇದ್ರಲ್ಲಿ ಏನಾರ ಜಾಯ್ನ್ ಆಗ
ಏನಾದ್ರು ಅಟೆಂಡೀಸ್ ಲಿಸ್ಟ್ ಹೋದ್ರಾ?
ಹಾ, ಓಪನ್ ವೆಬ್ಎಕ್ಸ್ ಮಾಡಿ ಸರ್. ಓಕೆ, ಇಲ್ಲ ಇನ್ನೂ ಇಲ್ಲ, ಅಲ್ಲಿ ನಿಮ್ಗೆ ಪ್ರೋಸೆಸ್ ಆಯ್ತಾ ಇದ್ರೆ ಅದು ಸುತ್ತಾ ಇರುತ್ತೆ ಸರ್, ಸರ್ಕಲ್ ಅದು. ಉಮೇಶ್ ಸರ್, ಹಲೋ.
ಹೇಳಿ ಹೇಳಿ, ಏನು ಎರರ್ ಬರ್ತಾ ಇದೆ ಅಂತ ಅಲ್ಲಿ?
ಹಾ, ಜಾಯಿನ್ ಅಂತ ಗೊತ್ತಾಯ್ತಂತ. ಈಗ ಏನು, ಈಗ ಲಾಗೌಟ್ ಆಗ್ಬಿಟ್ಟೆ ಮಾಡಿ ಸರ್ ಇನ್ನೊಂದ್ ಸರಿ.
ಲಾಗೌಟ್ ಆಗ್ಬಿಟ್ಟು ಮಾಡ್ಲಿ.
ಎಸ್ ಸರ್, ಎಸ್ ಸರ್, ಎಸ್.
ಇಲ್ಲಾಂದ್ರೆ ಸಿಸ್ಟಮ್ ರೀಸ್ಟಾರ್ಟ್ ಮಾಡಕ್ ಹೇಳಿ ಉತ್ತಮ.
ಇಲ್ಲ ಇಲ್ಲ, ಈಗ ಲಾಗೌಟ್ ಮಾಡಾಗ್ಲಿ ಬನ್ನಿ ಸರ್, ಮೋಸ್ಟ್ಲಿ ಆಗ್ಬೋದು. ಸರ್ ಆಗ್ಲಿ ತೊಂದರೆ ಇಲ್ಲ, ಮಾಡಿ. ನೇತ್ರಾವತಿ ಮೇಡಮ್ ಅವರೇ, ಹಲೋ. ನೇತ್ರಾವತಿ ಮೇಡಮ್,
ಸರ್.
ನಿಮ್ಮ ಕ್ವಶನ್ಸ್ಗೆ ಸರ್ ಲಾಸ್ಟ್ ಅಲ್ಲಿ ನಿಮಗೆ ಅದು ಆನ್ಸರ್ ಮಾಡ್ತಾರೆ ಮೇಡಮ್. ಒಂದು ಸ್ವಲ್ಪ ಟೆಕ್ನಿಕಲ್ ಇಶ್ಯೂಸ್ ಆಯ್ತು, ಸ್ವಲ್ಪ ಜಾಯ್ನ್ ಆಗ್ತಾ ಇದಾರೆ ಅಗೇನ್.
ಸರಿ ಸರ್, ಅದ್ ನಾನು ನಿಮ್ ಇದ್ರ ಹತ್ರನೇ ಇದೆ, KST ಹತ್ರನೇ ಇದೆ, ಹೋಗ್ತಾ ಇದ್ದೆ ಈಗ.
ಓ ಹೌದಾ?
ಸರ್ ಇದು ಬಸ್ಸಲ್ಲಿ ಹೋಗ್ತಾ ಇದೀನಿ ಸರ್, ಡಿಸ್ಟರ್ಬೆನ್ಸ್ ಇದೆ.
ಇರ್ಲಿ. ಓಕೆ, ಸಮ್ ಮೋರ್ ಮಿನಿಟ್ಸ್ ವೇಟ್ ಪ್ಲೀಸ್.
ಸರ್ ಅವ್ರು PPT ಕೊಡಕ್ಕಾಗುತ್ತೆ ನಮ್ಗೆ? ಅವ್ರದ್ದು PPT ತುಂಬಾ ಚೆನ್ನಾಗಿತ್ತು, ಸಿಂಪಲ್ ಆಗಿತ್ತು.
ಕಳ್ಸ್ಕೊಡ್ತೀನಿ, ಮುಗಿದ್ಮೇಲೆ ಎಂಡ್ ಆಫ್ ದಿ ಕಳ್ಸ್ಕೊಡ್ತೀನಿ.
ಸರಿ ಸರ್, ಓಕೆ.
ಸರ್ ಮೊದ್ಲಾಗಿದ್ದೀರಾ ಸರ್ ಜಾಯಿನ್ ನೀವು? ಅದು ಹೌದಾ? ಉಮೇಶ್ ಸರ್
ಹೇಳಿ
ಸರ್, ಹೇಳಿ ಏನಾಗಿದೆ ಹೇಳಿ ಸರ್. ಇವಾಗ ಸರ್ ಅನ್ಮ್ಯೂಟ್ ಮಾಡಕ್ ಆಗ್ತಾ ಇಲ್ಲ ಅಂತಿದ್ರಿ ಸರ್.
ಒಂದ್ ನಿಮಿಷ, ಅವರು ಇದ್ರಲ್ಲಿ ಇದಾರ ನೋಡಿ, ಅವರು ಅಟೆಂಡೆನ್ಸ್ ಲಿಸ್ಟ್ ಅಲ್ಲಿ ಹೋಗಿದ್ದಾರೆ, ಪೆನಲಿಸ್ಟ್ ಹಾಕಿ ಅವ್ರನ್ನ.
ಅಲ್ಲ ನೋಡ್ತಾ ಇದೀನಿ, ಇಲ್ಲಿ ಅಟೆಂಡೆನ್ಸ್ ಕಾಣ್ತಾ ಇಲ್ಲ ಅವ್ರ ಹೆಸರು.
ಇಲ್ಲ, ಗಣೇಶ ಅಂತ ಆದ್ಮೇಲೆ ಹರಪ್ರಸಾದ್ ಸರ್ದ್ ಇದೆ ನೋಡಿ.
ಒಂದ್ ನಿಮಿಷ ಸರ್, ಹಂಗೇರಿ ಸರ್. ಎಸ್ ಸರ್, ಇರಿ ಸರ್, ಮಾಡ್ತೀನಿ 1 ಮಿನಿಟ್ ಸರ್ ಪ್ಲೀಸ್. ನೋಡ್ತಾ ಇದೀನಿ ಸರ್. ಉಮೇಶ್ ಸರ್ ಯಾರಾದ್ಮೇಲೆ ಇದೆ ಅದು?
ಫಸ್ಟ್ ಇದೆ ನೋಡಿ.
ಫಸ್ಟ್ ಸ್ಟಾರ್ಟಿಂಗ್ ಇದೆಯಾ? ಒಂದ್ ನಿಮಿಷ ಸರ್, ಒಂದ್ ನಿಮಿಷ ಪ್ಲೀಸ್. Now we can join, sir.
Now I can
PPT share ಮಾಡ್ಕೊಳ್ಳಿ ಸರ್.
start video also. So PPT 1 ನಿಮಿಷ share ಮಾಡ್ತೀನಿ.
ಇಲ್ಲೇ ಮಾತಾಡಿ ಸರ್ ಡೈರೆಕ್ಟ್ ಆಗಿ, ನೋ ಪ್ರಾಬ್ಲಮ್.
Okay. Yeah. Okay.
ಇಲ್ಲೇ ಕೊಡ್ಲಾ? ಕೊಟ್ಟಿದ್ದೀನಿ.
ಶೇರ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ ಈಗ.
ಸರ್ ಪ್ಲೀಸ್ ಸರ್. ನೆಟ್ವರ್ಕ್ ಇಶ್ಯೂ ಆಯ್ತಾ ಸರ್? ಇಲ್ಲ ನಮ್ಮಿಂದ ಏನಾದ್ರು
ನೆಟ್ವರ್ಕ್ ಇಶ್ಯೂ ಅಲ್ಲ, ಅದೇನೋ
ಸಡನ್ ಆಗಿ ಡಿಸ್ಕನೆಕ್ಟ್ ಆಗ್ಬಿಟ್ಟಿದೆ.
ಓಕೆ ತೊಂದರೆ ಇಲ್ಲ ಸರ್.
ನೆಟ್ವರ್ಕ್ ಕರೆಕ್ಟ್ ಆಗಿದೆ, ಇಲ್ಲಿ ಕನೆಕ್ಷನ್ ಎಲ್ಲ ಕರೆಕ್ಟ್ ಆಗಿದೆ, ಏನು ತೊಂದರೆ ಇಲ್ಲ. ಬರ್ತಾ ಇದೆ ಅಲ್ವಾ ಈಗ?
ಎಸ್ ಸರ್ ಥ್ಯಾಂಕ್ ಯು ಸರ್.
ಲೆಟ್ ಮಿ ಕಂಟಿನ್ಯೂ.
ಎಸ್ ಸರ್ ಪ್ಲೀಸ್, ಥ್ಯಾಂಕ್ ಯು ಸರ್.
So sorry for the technical glitches we have encountered. I'm sorry for it. We are talking about the applications of recombinant DNA technology, and to go further, I was talking about engineered antibiotics. So antibiotics, as you all know, have varied applications that have helped us survive all the challenges which the microorganisms poses us with by causing diseases. So antibiotics produced by microorganisms are very effective against different viral, bacterial, protozoan diseases. And some of the popular ones which you know are tetracycline, penicillin, streptomycin, novoviricin, bactericin, etc. And what is recombinant DNA technology has to do with this? The recombinant DNA technology has helped mankind in increasing the production of these antibiotics by actually providing us with the tool or with the provision of improving the microbial strain through modification of genetic characteristics. Thereby, they produce more antibiotics or more effective antibiotics. Now, it doesn't stop there. We have gone forward, and other challenge which we face is diagnosis of diseases and preventing those diagnosed diseases. Recombinant DNA technology here also have helped us. That is, they have greatly solved the problem of conventional methods which we use for diagnosis of diseases. It provides methods for the prevention of a number of diseases like AIDS, cholera, etc. And monoclonal antibodies are very useful tools in the process of disease diagnosis. So various diseases, we have developed monoclonal antibodies which have rendered a technique for us to detect the diseases faster and better and more specific, with more specificity. Of course, earlier I mentioned gene therapy. Gene therapy is undoubtedly the most beneficial area of genetic engineering for human beings. This technique is a magic wand we have with us. It is the treatment of diseases by transfer and expression of genes into the patient's cells so as to ensure the restoration of normal cellular activity from a condition wherein we had a defective gene corrected by gene therapy and a correct gene is inserted and made functional, so that what problem the person was encountering with a defective gene is corrected. And this technique of gene therapy has provided new hopes for mankind in attempting various different kinds of genetic disorders. Forensic applications. Recombinant DNA technology is so vivid and it is so widespread in its application. You find its applications in forensic sciences. The forensic sciences which largely rely on a unique technique which we all know today as DNA fingerprinting. It is a great tool for identifying any person by analyzing their only unique identity, that is their DNA. And this can be done from any source. This can be done from any source of DNA from human beings. So DNA fingerprinting also helps to solve the problems of parentage and to identify criminals based on their identity, based on the technique of DNA fingerprinting, identifying very specifically whom the DNA belongs to. Environmental protection is an area where recombinant DNA technology also finds application. Genetic engineering makes its contributions to the environment protection in different ways. Most important to mention are the new approaches here utilized for waste treatment and bioremediation. Major approach in environment protection use the use of recombinant DNA technology for degradation of toxic pollutants which harm the environment. How actually this can be done? You have seen various sci-fi movies wherein microorganisms can be modified to eat up or to digest various things which they naturally cannot do. This can be rendered by a series of research efforts which can lead to generation of such recombinants or recombinant microorganisms which produces enzymes which are actually capable of degrading, which naturally cannot be degraded by the natural enzymes which these organisms produce. So, like for example, non-biodegradable chemicals, plastics, and also to various extent, various different kinds of chemicals or various different kinds of materials which are not naturally degraded in the environment. Recombinant DNA technology has covered us with generating such organisms, producing such different kinds of enzymes, proteins, and molecules which have the capability to degrade it in various different manners. So we have been able to provide answers for non-biodegradable forms of waste materials and also toxic waste. Biological energy production is another area wherein recombinant DNA technology has ventured out into. Recombinant DNA technology essentially plays an important role in beneficial and large-scale production of biofuels like biogas, biohydrogen, biodiesel, bioethanol, etc. The list goes on. Genetic engineering here helps to improve organisms for obtaining higher product yields and product tolerance also. Because what really happens, these are all produced by microorganisms, modified microorganisms. A important feature in all these organisms is whenever there is a higher production of the byproducts, the organisms tend to recognize this and retard its growth. So hence, we have been also able to, mankind has also been able to generate such transformed organisms which can tolerate higher concentration of these so-called toxic chemicals for the organism itself. Genetically stable, high-producing microorganisms are being developed using recombinant DNA technique, which aid in an efficient production of bioenergy. Now, let us look into, in a wider sense, what the recombinant DNA market holds for us. The growth of the recombinant DNA technology market is being driven by the increasing demand for biopharmaceuticals. The rising prevalence of chronic diseases leads us to look into various different paths and the technological advancements which we are making in the recombinant DNA technology itself. There is a rising prevalence of chronic diseases, which is another major driver of the market growth in recombinant DNA technology. And we see that there are many key players like Thermo Fisher Scientific, Inc., Kaizen, GeneScript Biotech Corporation, New England Biolabs, and Merck Labs. And apart from that, various Indian companies have also surfaced during this period of COVID. Technological advancements in recombinant DNA technology are also contributing to the growth of the market. What are the limitations of recombinant DNA technology? In spite of having all such wide applications and a technology which has rendered such important tools in our hands, there are certain concerns also. The major concern which we are all worried about is destruction of native species in the environment. Genetically modified species are introduced in. So will this be a challenge? Will this microorganism which we have recombined destroy all the naturally existing organisms? Resilient plants can theoretically give rise to resilient weeds also. And this resilient weed can be very difficult to control. Cross-contamination and migration of proprietary DNA between organisms, recombinant organisms contaminating the natural environment, and recombinant organisms are population of clones which are vulnerable in exact same ways. And a single disease or pest can wipe out the entire population quickly because here It is rendered because of the recombinant DNA technology. It is then the gene which is or that clone is also vulnerable in the exact same way as its parent. So hence, one infection can wipe out the whole population. So creation of superbug is also hypothesized. So are all these concerns which can we actually uh, meet up to these challenges, or are these really threats wherein we do not have any answers? That is a big question to answer for the mankind. Hopefully, we will be able to surpass or understand this technology better and wipe out all these concerns, which some common concerns have listed out here. Apart from all this, above all this, we have one important question: ethical concern about humans trying to play God and mess with the nature's way of selection. It is exaggerated by the fear of unknown of what all can be created using the technology and how it is going to impact civilization. And in such a situation, the system might lead to people having their own genetic information stolen and used without permission. So all these are, this is another area of concern wherein it is very difficult to decide, are we going to restrict or are we going to have a hold on these kind of ethical issues? But however, recombinant DNA technology has provided us a lot of advantages and such technological developments which would never have been possible without this technology. This is what I said. I did not put in the content page. Everybody today is talking of artificial intelligence. Now, in the last few statements which I have to make, here we see that there is widespread success exemplified by innovations like one AI tool called as ChatGPT, which has captured the attention of everybody, and everybody talks about ChatGPT today or the applications of artificial intelligence in many different areas. Likewise, in life sciences also, on to the human being, lives of human beings. So combining the advancements in biotechnology like recombinant DNA technology with artificial intelligence leads to an unprecedented potential for arriving at novel solutions for all the challenges we are having today. So this convergence actually holds promise for addressing numerous global challenges and aligning with critical sustainable development goals. Notable areas wherein recombinant DNA technology and combination of recombinant DNA technology and AI can lead us into food security, health and well-being, clean water for everybody, clean energy, responsible consumption and production, climate action, life below water, as well as all those endeavors to safeguard, rehabilitate, and promote the sustainable utilization of terrestrial ecosystems we have. So, artificial intelligence is now pervasive in the realm of life sciences, spanning diverse domains such as machine learning, big data analytics, knowledge discovery, biomedical ontology, knowledge based reasoning, and natural language processing, decision support, reasoning under uncertainty, temporal and spatial representations and inference, and methodological facets of explainable artificial intelligence. These are the advancements in artificial intelligence, which are accompanied by applications of biotechnology and thus illustrating their far-reaching potential for a future we can never expect. And what door it opens for the mankind is still not unknown. It is a very strong tool, very impressive tool which we can utilize, combining one on the other end artificial intelligence, the other on the other end recombinant DNA technology. So where we are leading to, we don't know, but we are sure it is for a better and brighter future with less challenges for the mankind to face. Thank you. This is my email, and you know how to get in touch with me. So at the end, before I end my lecture, I once again thank Dr. Ayappan, Dr. Ramesh, Vijay Gowda, and the whole team of everybody at KSTA for having provided me this opportunity and offering my salutations to His Holiness Jagadguru Shri Shivaratri Deshikendra Mahaswamiji, who has groomed me into contributing towards knowledge development in our state and also on a wider sense to the younger generation of our nation. Thank you, Ananda.
Thank you, sir.
Thank you, sir.
Sir, somebody asking ಇದು PPT ನ ಶೇರ್ ಮಾಡಕ್ಕಾಗತ್ತಾ ಅಂತ ಕೇಳ್ತಾ ಇದ್ದಾರೆ ಸ್ಟೂಡೆಂಟ್ಸ್ ಮತ್ತೆ ಕೆಲವೊಂದು
ಮಾಡೋಣ ಸರ್.
ಲೆಕ್ಚರರ್ಸ್. ದಯವಿಟ್ಟು ನನಗೆ
anyway it's on the YouTube, they can watch anytime, right? You have put it up on the YouTube.
ಎಸ್ ಸರ್. ನನಗೆ ಶೇರ್ ಮಾಡಿದ್ರೆ ಸರ್ ವಾಟ್ಸಪ್ ಅಲ್ಲಿ ನಾನು ಗ್ರೂಪ್ ಹಾಕೊಡ್ತೀನಿ.
ಮಾಡ್ತೀನಿ ಸರ್, ಮಾಡ್ತೀನಿ, but only with a official request not to use it for any other purpose other than education.
Please sir. Thank you.
Sure sir, definitely share.
Okay sir.
Thank you very much, sir. At the outset, we would like to thank you once again for delivering today's session on behalf of KSTA and on behalf of all the students and teaching faculty. We once again thank you very much, sir.
Thank you.
Sir, one minute, sir. 1 minute, sir.
Yeah, yes, please.
Sir, one question is there.
Yes, sir.
Can you just elaborate on electroporation and one question is there?
Yeah, electroporation is another technique used to introduce in the process of recombinant DNA technology. Electroporation is another technique to introduce the recombinant DNA into the host cell, wherein electric pulses are used to create a hole in the cell, a temporary hole in the cell, and inject the required DNA fragment into the host cell, and then it takes care of the process of integration and ligation.
And one more question.
Another technique for transformation.
And one more question, sir.
Yes, sir.
Please explain about chromogenic substrates, sir.
Actually, chromogenic substrates in this context, I might not be able to elaborate, but I will be able to elaborate on a different occasion now because it's not related all that to this topic of recombinant DNA technology. We will definitely deal with that. I can provide you details at a different occasion.
Thank you, sir.
With students, few of the teachers also have joined. It will be helpful for them also. These are the questions from them also, sir.
Yeah. We will do that. No
Thank you, sir. Thank you, sir.
Thank you very much.
If there are any other questions, please contact us from the email or through the call or through the WhatsApp group. We will get back to your queries through the answers from the sir. And meanwhile, over to you, Siddhegoda.
Thank you, Amish.
Thank you, sir.
Thank you, Siddhegoda. Thank you.
So today we are giving a very informative talk on recombinant studies, sir.
Yeah.
So once again, on behalf of KST and myself, thank you, sir.
Thank you very much. So can I exit?
Yes, sir. Yes,
Thank
sir.
you, sir. Sir,
So,
we will be contacting you for future programs also.
Definitely, sir. Definitely.
If any physical programs we would like to organize here.
Most happy to attend. I'll be most happy.
Thank you, sir. Thank you, sir. Thank you once again. Thank you very much. Yes.